what if we could build the world's first programmable binder to these disordered regions, like a CRISPR, but for proteins? So given a sequence of amino acids, you're building a binder to these, and you can precisely and selectively and potently target them. That would be a game changer. And the vision was, today we don't think about building a PCR primer. You know the rules of how the PCR priming works. Think about all the innovations that happen. You just go and design it. What if you could create a similar system where you could make a binder to any disordered region by just knowing the sequence?
This is the Nucleate Podcast, the voice of the next generation of biotech leaders.
Welcome to the Nucleate Podcast. I'm Anastasia and I'll be your host today. Today we are speaking with Eswar Iyer, co-founder and CEO of Aikium, a therapeutics company working to solve one of the biggest challenges in drug development, targeting undruggable proteins, those parts of the proteome that can be reached by traditional small molecules or antibodies. Aikium's approach combines AI with high-throughput wet lab screening to explore a vast space of protein interactions, going far beyond what's been experimentally possible until now. Their platform, YOTA-M², intelligently navigates trillions of potential proteins binders to design precise, functional therapeutics for previously unreachable disease targets. Eswar brings a rare combination of scientific depth and technical innovation to this work. He holds over 100 patents, earned his PhD in molecular biology at George Mason University, and completed his postdoc with George Church at Harvard Smith Institute. Before founding Aikium, he also helped lead platform strategy and acquisitions at NX Genomics, driving innovation at the forefront of biotech. In this episode, we dive into what it takes to build a company around a hard problem, from imposter syndrome and failure to perseverance and building the right team around you. Enjoy the episode. Thanks for joining Nucleate Podcast, Eswar. We are looking forward to the conversation to learn about yourself, your journey, and also the company you're building.
Great. Thank you so much, Anastasia. It's a pleasure to be on the Nucleate Podcast and looking forward to having this conversation.
Great. Before we dig into the hard science, let's start with your childhood and also the education and your path that brought you where you are right now. So could you share with us where you are from, your upbringings, what your parents did, and also if you envision a career in science from the early on, or if there was some kind of fate that brought you to science?
I was always curious. So I was the kind of kid who liked taking things apart at home. And, I always, if I found an animal that was injured, I'll bring it home. I was just very curious and passionate about things that were happening in the real world to understand it. So as a result, I was not very good at school. So there was a balance. I was trying to make useful things. I think people started calling me a young scientist. That's when I realized, scientist was a career. I didn't know these were things. And they would call me an inventor, and I realized, maybe that's what I want to do. So it just was a very natural tendency that happened from childhood. And then I realized I wanted to be a scientist / inventor. But then I realized that in order to do that, somewhere down the line, I was not good in academia because I was very engrossed in things outside. And I realized sometime that you need to do a PhD in order to be any of these things. So then I became very hyper-focused to get into a PhD. So I think that's sort of what sparked my curiosity. And of course, my father was also an academic and an engineer. So sometimes I would browse through his books and find drawings. And I always felt very compelled to build things that were reading was not very satisfactory to me. I felt like I had to experience it. So if I was reading about how to build, breaking hydrogen and oxygen from water by passing electricity, I had to build it and, do an explosion myself. There are no YouTube there. So I just would get obsessed about doing those things. So that was really where I got my interest in science and tinkering and building things.
That's super interesting, especially being called a young scientist and also trying the experiments yourself at home. I hope you didn't have to renovate a lot because of the failures from experiments, or it's like some stuff that didn't go as planned.
I'll share one or two funny incidents, but I think I've come close to electrocuting myself close to 5 times. I think the idea was, I thought if a motor runs, and this was like when I was in my 3rd or 4th grade, a small motor runs on 1.5 volts. But I thought, well, if you connect it to the main power, it could be like a turbine. And I thought it was a great idea, and I connected it with my hands. And then I basically got a huge shock. But I was more worried that my mother will find out. So I had to make sure that, all evidence was cleared. I also been bitten in my quest for rescuing animals by dog, cat, I think a crow, a bat once. So I just was very curious, and I don't wish this on any of my son. I'm very worried that he carries some of these tendencies, but yeah.
That's a great, kind of funny story. In like looking back to it, I think in the moment it wasn't as funny as it is now. But yeah, but it's a very interesting childhood that you had. And you also mentioned your PhD, and you're kind of, at some point you knew, oh, I want to become a scientist / inventor. So was your plan to stay in academia and to do the academic road, or did you know then that you wanted to kind of go on and start your own company?
Yeah, I did not know I wanted to again start a company. I just knew, I did not know what would be the best outcome for my skills were. I just knew that there are certain things that you do without thinking, without making an effort. It just happens naturally. So my addiction was tinkering, understanding, and building, just curiosity. So I could not resist it. And so after my schooling, my undergrad was at a place called BITS Pilani, which is in India, in Rajasthan. It's a very nice school. But I was again not very focused on academia. But every year we had a science competition. So I was very obsessed about doing those well, and I got a few awards that encouraged me and kept my hope alive that maybe I could do something. And then I got into PhD program after that. Again, I did not apply for any jobs. I guess looking back, it was a very risky from a career perspective. It was almost foolhardy that I did not apply for any jobs or so. I just had this feeling that everything will work itself out, even though my academics grades were pretty low. But George Mason University seemed to have, taken some interest, given me an admission. And that's really where things changed. So I worked with a professor called Daniel Cox. He was a new PI. He's working on developmental biology and neuroscience. I think that, joining there just changed everything. My academia became better. Now everybody rewarded me for the tinkering that I was doing. So it was a very interesting change. I think, so the question comes back to, did I know I wanted to do startup? No, I still was going after very basic biology. And because my mentors surrounding me were all in academia, I felt like I wanted to be in academia and I wanted to be a professor of sorts. I did not know that is really what was my surrounding, and I kind of took on that aspiration. But as I was doing things, I realized that by the end of my PhD, that I wanted to do something more applied. And maybe there's a chance that it could be working in industry. I did not even at the time clearly articulate, that I wanted to do a startup. I thought maybe I would do a startup eventually or sorts. So it was a journey because I was always building these tools. Like once we did not have a part for a microscope, and I had made this deal with somebody at the machine shop to teach me how to machine things if I helped clean the machine shop. His name is Sander Neyers. He's running the Machine Shop Inventors Club. And in the process, I would go and build the parts that I need and come back and do things. So I knew I had to convert this into something meaningful. So that led to my next.
Yeah, that's very interesting. Also, very hands-on approach, learning a lot and applying it directly. And you meant that this led you to the next thing, which would be working in the George Church lab at Harvard. No.
Yeah.
Yes.
There was a small thing in between, but yes, roughly.
What was the small thing? Now we're curious.
All right. So after I finished my PhD, when I was finishing a PhD, and perhaps this will be the most relevant part for the graduate students listening here, there was not a good plan. I thought I wanted to apply to industry, and I applied to industry jobs, but it was not really a good fit, or I didn't really hear back. Maybe industry was not developed. Maybe I was not doing a good job. But I did that really as a backup because I was told I should apply for something. But what I was really passionate about was to solve a big problem. I was thinking, if I were not worried about publications. Academia gets obsessed about publications. I thought if I was not obsessed about worrying about publications and things, if I could do the riskiest thing that I could do, because I felt like in my life, I had already become successful. Because I did not mention this, but I failed my 9th grade before. So I was so bad in academia that I failed my 9th grade. So when I actually succeeded, finished my undergrad, I already felt like I had, gone above and beyond what was, what I expected out of myself. So I thought if I could take a risky, I've already failed quite a bit. If I could take a really risky approach, what is the big challenge I would solve? So then I thought, neuroscience was backing out from drug discovery at the time. I heard a lot of pharma companies backing out because you could not reproduce it. People are using mice or rats for testing. And I thought, if only I could build a tiny human brain on a microfluidic chip, like take a stem cell, convert it into a tiny human brain or brain-like tissue that was reproducible in large number. And then I could test different compounds and get some quantitative readout from it. I thought it'd be a game changer. So I got very, this vision was so compelling that I could not stop thinking about this. And eventually led to writing an email to the director of Wyss Institute at Harvard, Professor Don Ingber. I wrote to him saying, I have this idea to build a brain on a chip. I think this is what I'll do. And after I sent the email, I knew I would not ever hear back from him. So I felt like, then why did I send it? But then I felt like I had to do my part and I forgot about this. And this is really when the universe conspires, when you really have, I felt like the universe conspired to get me there, because I met one of my mentors later in that week, Dr. Lance Liotta, who invented this technique called laser capture microdissection. And turns out he knew Professor Don Ingber, and he said, he made a recommendation. As a result, my resume was seen, and then I was invited, and then I got a job at the Harvard School of Engineering and Applied Sciences, building a human brain on a chip. And my job was to build a human amygdala. So I worked there for a short time. And there I got trained on nanofabrication, microfabrication, machining, growing cells of different types, and so on. So that was my job before I went to George Church's lab. And then I'll tell you how I went to George Church's lab, because that's the next question perhaps that you asked. I was working there for about a year, but I realized that perhaps I had this insight that the way we think about engineering is not the same as biology thinks about engineering. Biology is self-assembled and organized. From a single cell, you form a whole organism that's functional. Whereas the principles of engineering have not yet caught up fully yet. We still try to perhaps print and put things together in a way. And I felt like if I could only discover the basic, starting points to build a human brain, I could design a human brain and build it. I could print the right gradients and so on. So around this time, I was walking at the street of Kendall Square, and I saw Professor George Church walking by. And I struck up a conversation, started talking, and we walked for quite a distance. And, ended up being a sort of interview. And, he invited me to the lab to work on things. And that was the best thing that ever happened, the most serendipitous, interesting thing. So that's how I went to George Church's lab. And there I worked on exactly that. I took, stem cells that are derived from his skin cells, PGP1, tried to figure out how to make organoids, what are the challenges, across the board, and really focused on trying to solve some of those problems.
That's a very interesting story, especially this thread of you taking risks and being rewarded afterwards, which is also a great lesson for our listeners. So basically, you need to be bold, you need to go out there, and also if you have an idea, be bold and take the risk to actually see if something great comes out of it.
And I would say, maybe add one small note here. In the near term, it will almost look like a failure. My term at Harvard, the 5 years, 1 year at School of Engineering and with 4 years with George, every day felt like I was on the verge of failure. So this is almost an 8-year cycle, 8 to 10 year cycle, when things that look, utterly failing feel like great successes behind. So I just want to add that perspective, because I did not know that. I was not sure whether I was in the right path. I just knew I was compelled to do this. I couldn't do anything else, but I was not sure whether it will lead to anything meaningful in the near term or long term, or whether I was differentiating myself strategically into, useful career or not. So it looks like it was bold, but it could be easily defined as also being slightly foolish. And you would only know this after a 7 to 8 year period.
Yeah, that's also a great insight that ultimately in the end, things fall in place and you see, kind of the meaning of what you've been doing and also the result of it. So this being, kind of feeling like a failure, but afterwards finding out, oh, I've done something great and was the great decision at the time. So it's a very interesting insight also for our listeners. So you worked with George Church, who is a pioneer and also a very known name in the biotech space. What would you say were the best lessons that you took out of it? And generally, like, what are the most valuable lessons that academia can teach about real-world biotech?
Yeah, that's a great question. I'll answer the second part first. First, what does academia teach that can be useful for biotech? First, we need to prevent Theranos-like calamity. I think perhaps I would like to believe that it was started with a good intention, but so many things went wrong, and you need scientific rigor. So, academia teaches you that scientific rigor and lets you think boldly and lets you dream. Sometimes industry is pretty short term. It's derived from market forces that may be literally short term and so on. And there are trends that are fancy versus not. But sometimes you need to, art and science lead the space. Art sometimes imagine centuries before we can see things, and science can sort of imagine decades before and bring it to reality. And then industry can sort of imagine few years ahead and then sort of try to bring that into reality where the market is. You could also be too far ahead. So that's kind of how I would put these together. So in some ways, the mixture of art, science, and industry, they're all sort of related in that continuum. So academia teaches that, gives you the liberty on art and science. And here, George Church's lab is a great example of how that actually comes together. So what are the lessons I learned from George Church's lab? First, let me actually tell you what the Church lab looks like. Many people wonder how this lab functions. There's one person and there are 100 people. It's really amazing how George has put people together. He's really sort of assembled, partially self-assembled or sort of organized group of individuals who are very passionate and are looking for a purpose. And sometimes they may not have the best qualifications on paper, and sometimes they do, but he almost selects for people who are nice and are very passionate and want to sort of make an impact. And that kind of creates a cohort of people that's very interesting. In this group, you will also find artists. Like there's a resident artist called Joe Davis, who was an inspiration for me when I was there. And that really intrigued me and learned and grew in different ways. Then there was an ethicist, Jean-Tine Lunshoff, who was thinking about the ethical consequences of all the work that we are doing. It's quite a blend of people. And there are people who are getting involved in all of these. And George kind of was well-versed in these different spheres. So that is one aspect. And the second aspect is about the lab that is intangible that people don't realize. The amount of faith that George has on the people, how much confidence he puts in them. And in some ways, it transforms people. When you sometimes don't have faith in yourself, but someone has more faith in you, you kind of live through that experience. You sometimes don't see your potential. And it won't be obvious or apparent. But the fact that you're given an opportunity to work on something as big, again, you may feel like a failure every single day because you're not because of anything else, because you're striving, you have very high expectations and you want to do something meaningful, you want to have an impact, but it provides an opportunity. That's the kind of risk taking, it's nowhere. And that is the reason why so many things have, come out of that lab. And one of the most important thing that I think people don't realize fully that I would like them to know is today we have broken the Moore's law in sequencing costs from $1 billion a genome to, $1,000 and less. George was pioneering those innovations in 1980s, 1984, 87, where the concept of molecular multiplexing was laid that helped eventually break the Moore's law. So sometimes just thinking ahead, he thought about nanopore sequencing 1989, 90, and then it was patented 95, decades ahead. So being in a lab, which sort of was thinking that far ahead, puts a very high expectation on what you expect from yourself and also what you want to do. So what are the lessons I learned from George's lab? Well, I can distill it into few things that will be useful for the users. First was I got obsessed about this principle of why the Moore's law got broken. And if people are curious what I'm talking about, again, look up the cost of sequencing that went down. It was $1 billion when it started, and around 2005 or 6, they expected another 60 years before it will be a $1,000 genome. Moore's law is this law where every 2 years the price halves or so based on certain thing. But really, it's not a law. It's a trend that industry follows. But the Moore's law was broken. In 6 years, we achieved a $1,000 genome. The question is, I got obsessed in George's lab about what were the principles that led to breaking of Moore's law. And that can be reduced to 3 things. So that is really what I can sort of convey to people that, even if they forget everything else, maybe remember this. 3 things. One was miniaturization. You make things small. Second is parallelization. You do more of them side by side. But a lot of industries do this. The real reason why it was broken, the Moore's law, was multiplexing. You do 1 billion reactions in one chamber rather than do one reaction at a time. Think of sequencing chips. You do 1 billion reactions and then you take images and so on. And that is quite profound. If you think about it and you meditate on that process, you can apply it in many different ways. So I took an oath that I will work on any technology I work on will have these 3 principles. And since then, I have been working true to my promise, on those 3 principles: miniaturization, parallelization, multiplexing. How can you? So if anybody wants a 10x improvement thinking about things, you just have to apply these principles. It's simple, but yet it's complex. You just have to know how to apply them. What else did I learn from George's lab? Culture matters. How you treat people matters. It may not, again, it's an 8-year cycle. The best things that have happened to me came from things that I did not expect anything in return from. Just being nice to people, just working with people without expecting anything in return. And sometimes it's an 8-year cycle. It comes back in ways that you won't even anticipate. Sometimes someone you mentored will come back and mentor you after 7 years, or will make something happen when you least expect it. So kind of do things, do bold things, of course, do what is necessary to calculate whatever you need to do, but try to be generous and bold. And there's also this tendency of hiding your ideas. I notice people think their idea is so big and important that they don't want to share. And everything I learned in that lab is against that. It's not to say that to share with everybody indiscriminately, but find people that you trust and are competent and you want to share with them. They're not going to run away and take your idea and do things. And you may feel it's quite possible many of the intelligent people are thinking of the same idea as you. It's quite normal and natural. In fact, that's, if you have a good idea, in fact, it's not a good idea, And this is a quote that I'm using from Ed Boyden, who's also one of the mentors, where people are thinking about the idea. If it's a good idea, everybody's thinking about it. So you want to think 5 layers deep on what is the fundamental problem, why this area is not being solved. So work on hard problems, be generous, share your ideas thoughtfully with people you think will add. I think it builds a momentum of working with people. It sort of self-assembles into a momentum that is far beyond a single person working and trying to solve things. And any failure that you encounter working on really hard problems, no matter how passionately you poured in, it is going to come back. It's a 7 to 8 year cycle. You will reap its benefits, either in confidence or some other job or something else. There are tangible aspects of things. There are intangible aspects of things. If you don't see something tangible, you will have something intangible that will eventually convert into something tangible.
Yeah, those are great insights. So basically work on bold problems, be nice to people, and be open to with your ideas and share it and make it better with other people who are also as great as you, or sometimes even better. And there were also a lot of other insights that were great, but what struck me that you said about the right timing. So is it too early? Is it too late? My question is, how did you learn it in the lab? So did you have lab meetings where you're discussing your ideas or your experiments, and then you were talking about the commercialization potential of these, or how would you describe this process of learning, or like knowing when is the right time for it?
That is a great question. And so the question is, how do you know what is a good timing for the inventions or ideas that you're doing? And the honest answer was, I did not. And I was working on something that was very compelling to me. But that is how academia trains you to go on, work on things. However, in hindsight, after working in industry, there is a way to do it. There's a process to this. So if you are planning to be in industry, or if you're planning to do a startup, and if you're in grad school, sooner you know it, the better it is. If you're in a postdoc, sooner you know it, the better it is. And the process is as follows. There are two things. You either get lucky, you work on something that's becomes so interesting that then you want to start a company, and then you try to find a way into market. That is how a lot of these startups happen. There's nothing wrong in it, but there's a lot more of soul searching and finding the right fit of sorts. Sometimes it's too far ahead of its time. Sometimes it's not very clear. I would say sometimes maybe a better process is, if you know you're going to go into industry, is to think about the problems that exist and try to solve one problem that can also create a business that is sustainable. And finding that problem is not easy or trivial, but finding that problem is the most important thing. So work backwards from the market rather than try to fit in. This is an advice that many people give, but the actionable aspect of it is, the sooner you know it, start working on it and get involved with the kind of people who help you think about it. To me, it was such a jarring idea to, I always built useful things, just out of intuition of building useful things, but they may not always be the right format, product, or so for the market. So being part of things like Nucleate, Harvard Innovation Club, or other, there are so many resources online. Just understanding how do you understand market and how do you work backwards, and thinking in that framework will help you just identify problems that can become a big thing. And then you can take your science and shift it slightly, and then maybe find an application to that. So that is how you think about the timing. Of course, speak with a lot of people, learn how to interview people. Sometimes when you interview them, you give a pause after the question. You ask them, this is one question I like to ask. I ask them, if you had a magic wand to solve, to solve any problem, or it's very open-ended, to do X or Y, what would you do? And they would start talking, and they would talk about some obvious things first. But at some point, when there's a long pause, they'll usually give you a pearl of wisdom somewhere. And if you're able to gather that from enough people, I think there's something there.
That's also very great insight into actually trying to find a problem and not starting with the invention and trying to find the market, but trying to fit it into the market, even though it won't fit, or maybe you think that's very great company, but the rest of the people doesn't think like it, but more of going backwards and flipping this whole framework. How was the process for you? So you spent 4 years in your Charges lab, and what were your ideas? What was what did you think at this moment when you went off to basically 10x Genomics and then starting your own company?
Essentially, the process at George's lab was as follows. I think around the start of George's lab, I was still thinking whether I'll be in academia versus not, but I knew I wanted to do something useful. So I was optimizing always for having an impact in real world. I just did not know whether it's academia or industry, and all my role models seemed to be in academia. So I was thinking, hey, maybe I could have impact in real world by being people like this. But slowly I saw also mentors who are across, and then later, people who are in other, mostly in industry. So when I was in George's lab, I found a problem that I wanted to solve. The problem that I wanted to solve was as follows. My large vision was machine learning was slowly coming of age. The limitation that is going to be how do you generate data for biological systems so that you can eventually have a virtual embryo or a virtual organism. So you can model all these things. So when I worked backwards from that, I realized that I needed to build a method for rapidly perturbing biological systems and seeing what is the effect of that perturbation. So you make one small perturbation, read out what happened. Make perturbation, read out what happened. And if you could do this in large enough numbers, that you collect this data, the small bits of data that feed into a model, that will be useful. Now, I did not know anything about machine learning personally. I just knew what it was. I knew that field was coming, but I knew that the ability to read and write into biological system will be powerful. So that was compelling enough for me to, I did a lot of reading to sort of come to that conclusion. The second thing that I knew was applying the principles of miniaturization, parallelization, multiplexing. To gather this amount of data by standard methods would have taken a lot of money and a lot of cost. So I imagined an assay where you could do this in a pooled manner. So imagine if there's a dish of cells where you throw a bunch of guide RNA or small molecules, and each cells take up one of these perturbing agents. Then you read out, using some optical method, the phenotype of the cell, whether it got changed from disease to normal phenotype or so, and ask what went inside. And in that plate, you have 1,000, 10,000 perturbations or 100,000 perturbations of sort, almost like next-gen sequencing. So I called it, pooled optical image screen, or PLIS. We could have come up with a more creative name, but so I was, I kind of had a patent around this, and I wrapped a lot of things. I imagined all different variations of this, and I started working on this. I had no idea how big of a problem I was taking a bite off. I just knew I had to get started. So I started recruiting people. George would send some people down the line. George appreciated this problem. He said, he I think mentioned something like, congratulate you on your ability to choose a problem or something like that. But I felt very passionate. I started working on it. And I imagined that it would be maybe like in 6 months, I would solve some core aspect of it, like the readout problem. The challenge was, once you have something inside a cell, how do you read out what is happening? And a paper called Fluorescent in situ sequencing was just published in the lab in Science. I thought, oh, paper has been published. We have the know-how. And in 6 months, I'll sort of have that. And then I can put all of these things together. I was so wrong. It took me the next 3.5 years to actually solve that problem. And what we were trying to do is to detect of these guide RNA that's going in, which guide RNA has gone into which cell. So you needed a very sensitive method of detecting that. And that sensitivity was 1,000-fold more than what was published. You don't know any of this till you do it. And then you kind of realize each step. And you're failing so much. And then a company was spun out from George's lab. And a lot of people got hired into it. So from a team of 7 or 8, I became one. Mm-hmm. And I was working on everything from hardware, software, building the biochemistry. And also, this teaches you about focus. I did not know what it required to get this. I felt like I could wrap all of this together. And only when you do this and you fail, you realize how much more is to be done. In the end, I basically put all of this together into a paper called Bolaramis, barcoded oligonucleotides, ligated on RNA and amplified for multiplex in situ. Long name, but essentially it was the first.
Very long.
But essentially, we did meet the goal of detecting guide RNA that was 1,000-fold more sensitive than the previous method. But it took everything I had. It took everything I had, and I did not know that there was any future to this immediately or so. And around this time, actually, I have to say, I was almost ready to give up during the darkest hour. And this is when the mentorship of people that matter. George came to the lab meeting, and then he sort of came and talked to me and encouraged me. And he knew that I was now single from a 7 to 8-person team. And then he gave an advice. He said, when mention something like, you're good at this, I think you should continue doing this, because I had an offer to join another startup and so on. And I was like, why would I do this if it doesn't have any future? Everything is failing. And I was really burnt, like working, 14, 15, 16 hours a day. And it just didn't seem like there was anything coming out of it. And he said, I think this is what everybody is going to be doing. I think you're good at this. You should continue this. And when George said that, I think the best thing I credit myself for is having faith and saying, okay, I said no to the other opportunity. I basically doubled down and just worked on it. published that paper into bioRxiv. I knew the quality of the work, so I published it. 3 years later, we published it in peer review. And then, I was doing startup courses around the time. I was thinking maybe I can commercialize this in some way. I did not know how. There's already a company that was spun off from George Church's lab. I was thinking maybe there's some angle to this. I learned a lot from the startup courses. And here's another lesson I learned around this time. I thought industry was not a good thing. Industry is not something you should go to. And there was this company, 10x Genomics, which was sort of doing droplet-based things. And I was not as interested in the things that they were doing. But when I told George about this, he said, "Oh, it's a good company of some." He kind of endorsed it. I thought, oh, I should take this interview seriously. And it was the only company that I interviewed. Otherwise, I was going to do my own startup. Boy, I'm so glad I actually listened to it and interviewed. And turns out they wanted to go big on spatial omics. So it was just a perfect time for me to join, and I decided to join. It felt like a big risk because you don't know how industry is going to turn out. You're going from a very famous lab where you could be starting your own startup to now you're joining as a scientist in some company that you're not sure, but I think it was totally worth it. Perhaps that's the next thing that it comes to, but that's the next part of the story where I learned about how to build products in an industrial scale, which I did not fully appreciate in my previous role. And they both were complementary, and that came a whole circle. So that was the thing about timing that you asked, closing the point. How do you know about timing? You don't. You kind of talk with mentors and people, and you have faith in sometimes. I had faith in George. He said, this is what everybody is going to be doing. And I just dropped it and I did, and it became true for me.
Yeah, so those words where you're working for something and you don't see an end inside, and you're done with all your reserves are right now, like they aren't there, and basically it comes to those people who believe in you, who see your potential and encourage you to continue working and not giving up, because those kind of lowest moments are usually those that are close to the breakthrough or like close to the end.
You're absolutely right. I think that little encouragement made me realize that the choice I was going to make at the time will define who I'll be as a person. So I had a choice to either put my head down and solve it and finish it, or I could be the guy who does things all the way but does not finish it and then keeps doing that. And I didn't want to be that guy. I wanted to do it and finish it. So I made the choice. I'm glad I made the choice, and I'm glad that somebody gave me that push of encouragement and gave me the space to do it. And that made all the difference in the next sort of several years.
It's also great character trait of a CEO of a startup that's just starting out. So basically you were so stubborn at some point, or you're not willing to give up, that you, even though you have so many failures and you think that it will never work, that you still persevere and work through it.
That's a great point. And maybe I would even specifically say that as that evolution was going from external validation to seeking internal reasons for doing it. We often get lost in external validation, and when it's not there, sometimes we lose a lot of steam. But when you're in your utter failure and when you're in the worst moment, you still want to do it, and there's something compelling you, and you do it for reasons, whether to complete the thing or you believed it, or you want to follow the process, whatever it may be. It becomes more intrinsic. And in some ways that does make you perhaps externally stubborn or so, but it helps you get through it. The only way out is through. You can't have backups. And you just have to get through it. It's not glamorous, it's not fancy. It looks like that after 8 years, 7 years, but it is not.
It's a great insight. And we've been talking about starting a company and basically the perseverance that needs to it, the solving big problems. So I would guess now it's the time to talk about Aikium. And maybe you could share with us what Aikium does, and also the story and how you started it.
Sure. I'll do a brief bridge. I'll come out of Aikium by telling about my experience at industry, because I think that is important for this whole part to come together on how this fits. And I want to say this because I find many people, again, thinking about industry, but thinking it's a risk versus not, and so on. I highly recommend somebody to consider joining an industry and learning from it. I'm not aware of anybody who's regretted joining industry. Maybe it's a very biased cohort, but I think there's something you can learn. If you're only thinking about how do I become effective as a person on executing my idea and my visions so that my time is used usefully, or I'm become more effective at my craft, I think you can't go wrong in being challenged in industry or other things, whether it works out or not, if you have that positive attitude. So long story short, I joined 10x Genomics, and at the time 10x wanted to go big on spatial omics. I think the broad ask was, how do you go big on spatial omics? Which companies, how do you build products? What are the next big products? So in some ways, I was fortunate to be at the right place at the right time, and I helped architect a lot of that vision in the beginning at 10x Genomics. And in that process, I was deeply involved as a scientific advisor to 3 acquisitions, 2 from Sweden for $40 million each, and 1 from George Church's lab, which is Reed Cool, for $400 million each. And it's great because now I saw what it took to be on this side of the company to build products versus on that other side. And now the circle came complete. Once you acquired it, you realize that what I worked on, which I felt did not have any future, now is perhaps, was one of the first demonstrations of the kind of highly multiplexed reverse transcription feed technology that is coming together now. So it gives you a lot of confidence that, okay, I actually could see it and I could work on it. The second was I was working on multiple product lines. One of them, the Visium HD and Xenium. And each of those I contributed differently. For Visium, I contributed the workflow to make it seamless. One of the things I had contributed to was to see whether we can make, get rid of excess equipment and use PCRs just as an incubator and so on. There are these inserts that they sell, and just simplifying the workflow. Visium HD is their next generation product on high resolution microarrays. And this is one of the ones that I'm very proud of. I conceived, pitched, and de-risked the initial concept of this idea when I joined. And we were talking about this, and I made a prototype that I shared with the team, and they got really encouraged that, oh, we could do this. And then I had about a year of with this very small team and a brave team to de-risk this idea. In some ways, I want to say 10x, whatever it was doing at the time, this was one of the things that they did well, which they encouraged at least someone like me to take that seriously. And then that became a product line, Visium HD, which was a major investment from 10x Genomics. And it's a product line. And then for Xenium, I personally requested that I be put on the most challenging and complicated aspect of it, which was the decoding chemistry. Basically, to build a decoding chemistry, you have to be at the center of building a reagent that is imaging, and it has to be in the center of chemistry, biology. It has to be compatible with the cost, with the materials of the machining. You have to understand how the automation works, the software, how it's reading out, all of those things. And I kind of really thrived and enjoyed that. So I led the early decoding chemistry aspect of it and understanding what the error rates were and how it was coming and so on. The team was brilliant and amazing at 10x, and I think they are very good at product development. So these are things that you can see in the quality of the products that come out from 10x Genomics, which kind of speak for itself. So that was the experience that I had at 10x Genomics, and that is where perhaps a lot of the inventions also sort of came that are, perhaps more public right now. When I was done with 10x, I kind of knew, for the next thing that I wanted to do was something big in machine learning and protein engineering. Again, this feeling of something coming together. I was not sure what it was. And here, maybe I'll very specifically detail how I started thinking about building the company, if that is useful to anybody. The first principle that I said was...
Sorry, yes, but it's just a very interesting that you were always trying to work on the hardest problems.
On that, I'll share one principle and one epiphany. One principle that is useful is, what do you chase? What do you choose and what do you chase so that you're always aligned with the right thing? And one principle I got was if you're trying to chase a shadow, you can never catch a shadow. But if you turn around and then go behind, chase the source of light, the sun, shadow will automatically come behind you. So here the sun is the problem, the tough problem that's going to be meaningful for everybody around you, or it's a very challenging problem regardless. And the shadow is all the derivatives, money, fame, everything else that comes from it. So instead of chasing that, if you chase really tough problems, they should be derivatives that they'll come. So it keeps you focused on, when you have many things that you have to focus on, that's a very simple way to make a decision. So it's a principle. And the epiphany I had was, and this is something that you have at industry, I really wanted a promotion at one point. And when I got the promotion, I realized that I actually lost productivity for a few months, because I was thinking about the promotion, and when I got the promotion, I was happy. But the epiphany I had was, and I literally sat down and wrote pages of it. And I was wondering what happened to my motivation of things. I know I would come up with ideas all the time and fill this with notebooks. And I realized that sometimes HR is designed to expect as much performance out of you, not more, not less. That's really how things work. So if I truly wanted to do my best in a very limited time, and we all have very short times of productivity, some may be more fortunate to have more or less, to make the best of it, I could not worry about promotion or things. They're important for my career and so on and so forth. And I do need the money to sustain myself. But in some ways, it took away some very precious time for the best part of my productive life. So I kind of promised myself that I would not worry about promotions or titles. I'll try to do the best work, and they should sort of follow. So that was the epiphany I had.
Yeah, very interesting. So kind of not doing this career games, but focusing more on solving big problems. Yeah, like talking about big problems. So like your next big project was starting a company, and you wanted to share with us some steps on how you did it, which would also be very insightful for our listeners.
Great. I think I will share what I did and what we are doing. Time will tell whether this is the right approach or not. But I think my experience and age has taught me that this is the way to do for me. And this is how I'll continue to do. Maybe I'll evolve in some process. So I'll share it, whatever I have conviction in. So the broad idea was, again, machine learning and protein engineering is going to be important, because machine learning is certainly able to reason about complex things like proteins, their sequences, and so on and so forth. It's just starting to happen. So the question is then, what do we build? So now I'll break my own rule. I said, start with the problem. Now I had defined the problem broadly as rather as an opportunity as that space, but I did not know enough to know exactly what the problem was. But I knew before problem, there is one important aspect, which is people. Find the right people, and you'll figure out how to do the right things. So I was very focused. And again, this experience came from working with really great people in my PhD, my postdoc, and so on. I knew I had to find just the right people. And they also say you are the average of 5 people, that you hang out with. So you will be one of the averages. So I knew I had to find those people that are going to be passionate about solving a problem in the space. So I was fortunate to find Shankar, who was one of the best protein engineers that I know, and Venkat, who is one of the most brilliant, AI and computational scientists that I know. And we sort of came together. That's a different story on how we came together. But once we came together, we started thinking about the problem space. And the second principle was, if an idea is a good idea, it's usually not a good idea. It's a principle that I picked up earlier. I think a lot in terms of principles. So just try to think about principles and then follow them, because the world is very complex. It's hard to make decisions. So how do you find the problem? What is the fundamental challenge in drug discovery right now? That if you peel 5 layers is the bottleneck. And if you solve that one thing, it's worth your time taking a crack at it. Even if nothing happens, it's worth your time solving that problem. And if it's successful, it could be a game changer. It could make a huge difference to the space. And we started thinking about that. And that's when we realized there was a big elephant in the room. The elephant was all the technologies that we have, whether it's antibodies or small molecules, are biased, can only attack half the proteome, roughly 50% of the proteome are accessible by this technology. And the other 50% are just not accessible at all. It was not obvious. Nobody told us this problem, at least that obviously. And you read, and that's when you realize that these trends occur. And only because we had such a diverse team of passionate people who had united with the idea of doing our best. Our mission was very simple. We wanted to be useful. And we wanted the definition of the best job was where your greatest skill meets the greatest need. So we asked, is it building an instrument? Is it building a protein sequencer? Is it building this and that? And we said, if you could build therapeutics, that's going to be worth it, because you cannot prevent people from dying, perhaps, but you can influence their quality of life. And that is worth a purposeful career, purposeful life. So when we all aligned on that, that was basically, we said, okay, let's do it. How do we do this? And then we asked, what's the problem today? Why should we even start a company? Why are we any better than anybody else? That's when we saw the problem of IDRs. These disordered regions that roughly span 50% of proteome. You take any protein, it'll have these loops and tails that don't have a structure, and you can't bind anything there. And they're extremely important for the protein structure and function. And a large fraction of post-translational modifications are enriched in these areas, like phosphorylation, and that changes their hydrophobicity, and that changes the structure of the protein. So essentially, it influences this protein-protein interaction network. It's something that's so critical, so important, and yet so unreachable. Think about all the undruggable targets. Now we call them challenging targets, but undruggable targets, whether it's transcription factors or GPCRs and so on. This is one of the fundamental regions. Say GPCRs, the only accessible region is this little disordered region that's there. Most of it's buried in the lipid bilayer. Transcription factors, huge disorder regions. So he said, okay, what if we could build the world's first programmable binder to these disordered regions, like a CRISPR, but for proteins. So given a sequence of amino acids, you're building a binder to these, and you can precisely and selectively and potently target them. That would be a game changer. And the vision was, today we don't think about building a PCR primer. You know the rules of how the PCR priming works. Think about all the innovations that happen. You just go and design it. What if you could create a similar system where you could make a binder to any disordered region by just knowing the sequence? That would give you unprecedented programmability, modularity, and sort of precision on finding out, understanding the disease, and also targeting it. And that's kind of how we got started. And we basically assembled the company. In fact, Aikium means to come together in a sense. And here we thought we were building binders to targets. So they have to come together. But the bigger vision was people have to come together, leave their egos, and resonate with each other. So in some ways, the concentric, resonance pattern, that's kind of what it indicates. They need not be the same, but they have to be united on the goal, and they have to resonate, and they have to let go of their ego just a little bit to unite on the purpose. So that's those are the basic principles with which we got started.
That's a very interesting story, especially also the name, which is symbol for what you're doing in Aikium. Could you specify how you do it? So you have a new class of proteins, maybe a little bit more insights or details on what actually happens.
Sure. So we asked, okay, if you want to build a new therapeutic class of molecules, from engineering principles, what are the qualities of the therapeutic molecule that will eventually go into the human body? And we realized it has to be a human-derived protein, ideally, or a protein that has low risk of immunogenicity, that's ideally derived from a human proteome. And then it has to function the way we expect it to, bind to the disordered regions, modulate them in some way. And then they have to, of course, have favorable developability properties like stability, solubility, being monomeric, and so on. So with that, we started engineering, looked at a bunch of scaffolds in the human proteome, asked which ones would be useful. From that, we picked a few, started engineering them. And we started with a shoestring budget. Honestly, around the time was the worst time to get started. The market was crashing. Nobody wanted to invest in biotech, or it's very rare. And whatever was there, it was very risk averse. So this felt like a very risky proposition for anyone. So, but we're still very passionate. And so we got our first $50,000 check, we got started. We got the next $50,000 check, we got started. Next 100,000, we kind of kept doing that. So essentially what we built is we started engineering a human protein that starts binding this disordered region. In order to do that, we have built a platform called YOTA ML Square. It stands for YOTA scale machine learning with massive libraries. YOTA is 10 to the power of 24. The idea was, how do you optimize the protein-protein interface, binding interface? And here's another limitation that we encountered. A lot of the three-dimensional protein structures that we get from, say, ESM or other methods are trained on crystal structures. Crystal structures are derived perhaps in the not the most physiological state. In order to form a crystal, you screen a bunch of conditions, and in some conditions, you form a little salt-like crystal, and then you detect it. However, the crystallized regions are often the structured regions of the protein, and the disordered regions, you don't have a crystallized structure. Because they live in an ensemble, you can't crystallize them. So they're invisible almost in crystal structures. So we don't really have a good sense of how these structures even look to model them and so on. So it's a major challenge. In fact, even after the Nobel Prize in protein folding, there was a paper that was published that highlights 5 unsolved challenges in this space that's for protein design. And 2 of them are designing binders precisely, and the second is conformational flexibility of proteins, which broadly also encompasses the disordered regions. So what we realized was this is a data sparsity problem. If you could build more data on this protein-protein interface, then you would have an advantage. An algorithm is only going to be as effective as the data that you feed, and most of the data is somewhat stagnated or limited based on the data that that we currently have. So we spent time building a machine learning, a data pipeline which can generate massive amounts of data in a short time. That's the platform. So how does this work at a high level? Given a target, the algorithm, which we have patented and we have published on this unique algorithm, and we have several more that are developing, it evolves a protein that binds to the target computationally. However, the protein design is only as good as the data it's trained on. So what we do is we convert that into an experimental screening technique that we can screen in the lab. So upon click of a button, all the designs that are made computationally are converted into DNA libraries. These libraries are assembled at Aikium, where we just order short oligos, very large number of short oligos, and we assemble them in the right combinations so that we get the whole protein in the right variants and so on. That represents about 10 to the power of 12 to 10 to the power of 13 space. And the computational part represents 10 to the power of 8 to 10 to the power of 11 space. So they are multiplicative in some way. So the 10 to the power of 11 multiplies with 10 to the power of 13. That's when you get the YOTA, 10 to the power of 24 scale. So in other words, we are doing a multiplicative integration of AI and wet lab screening. So we can screen a larger parameter space rather than just look at one or the other. Then these DNA molecules are converted into barcoded protein molecules, where every protein is covalently barcoded with an RNA molecule. Then we had to actually improve this biochemistry quite a bit to make sure that the molecule is stable, selectable, can go through different cycles, the amplification. So there are many challenges to this. You literally think product development for a while. So we have this biochemistry that's highly optimized, which converts the DNA library into a barcoded protein library. And now we have a tube where 1 trillion molecules are sitting, AI designed molecules. So that's the uniqueness. These are all AI designed molecules, and there's 1 trillion of them. Then we can go and do positive and negative selection with your targets of interest, whether it's on beads or in different formats or virus-like particles and so on. Then you do selection. And once you do the selection, then using an optimized PCR reaction, you can amplify the barcode tags that led to the selection. So compared to the 1 trillion, you'll probably pull out, 1 million that likely bound. And then you repeat the selection process a couple of times, positive, negative, then you'll be left with a few. And when you sequence it, deep sequence it, you'll know which designs actually bound versus others. And that data can directly go into your algorithm for training. So the algorithm keeps getting smarter. So what we have built essentially at the core is a business of solving the data sparsity. And that is going to lead us to building better molecules. But this is going to be the limitation in the next few years. There's a lot of value in AI and perhaps a little bit of hype as well. But the fundamental, peel the layer, what's the fundamental bottleneck? Data sparsity problem. And that's kind of how this platform works.
Very interesting and also very understandable explanation. So thank you for that. So my question is, or maybe correct me if I'm wrong. So by having those multiple selection layers, you prevent this AI hallucination or those structures that are actually humanly not possible. Is it correct understanding?
That is right. So the AI is designing structures that human intuition might not have guessed, because you're now trying to get a very large, sometimes the binding interface can be 15, 18, 20 amino acids. And if you look at randomly select all of them and then screen the variants, it's 20 to the power of 20. It's a very astronomical space. Instead, the AI is allowing us to intelligently weight that parameter space and tells us, position 1, just look at these 5 amino acids, position 2, 3 amino acids, position 3, and so on and so forth. And we have the wet lab ability to precisely assemble that library, which we don't order. So all of that sort of comes together.
Okay, great. Looking forward. So IDRs don't only play a vital role in physiological process, but they also found in diseases such as cancer, neurodegeneration, and infectious diseases. So therefore, targeting those areas or like regions could open up like a new therapeutics avenues. But one of the problems is that they have sequence similarity to other proteins. How do you ensure that those binders that you make, they bind to the specifically to the regions that you aim for, and they do not disrupt the physiological processes in the cells?
That's a great question. So essentially, how do we achieve selectivity from making sure that they bind to other regions? So in some ways, based on the data that we have generated and assembled from various sources, we have a map of what these targets, what these proteins bind to. So we have some sense of what are their tendencies. Do they like, amino acids with, some hydrophobic, hydrophilic, or, mostly hydrophobic, or certain combinations of residues. So we have a very large catalog of these, preferences to different scaffolds and those combination paired binding. And that can be used for also doing this. But fundamentally, the problem is you're asking, many proteins have similar regions. How are you even going to target them? If I understand this right. And the answer to that is, because we are targeting in the sequence space, it allows us to choose pre-filter sequences that are unique by a few Hamming distances to other sequences. A Hamming distance is essentially if you have two sequences that are 15 amino acids similar, 15 amino acids long, if they are different by one position, that's one Hamming distance different. If it's position different by three positions, they're three Hamming distance difference. It's a terminology that has been borrowed loosely from the DNA space. So if you can always select for sequences that are pretty unique to that region. Here's a great example. GPCRs are extremely challenging to make binders to, or antibodies to. There are 3 approved antibodies for GPCRs because they're all targeting certain regions on the exposed part, which is challenging. But what's unique is most GPCRs have an N-terminal region that's loosely hanging around, that's accessible. And that N-terminal region is not very conserved. So it's almost unique to every GPCR. So it's almost a barcode for every GPCR. So you can go after the unique regions in the GPCR and allowing you to do it. And that's kind of how we went after the chemokine space and so on. So long story short, we do it in both ways, or 3 different ways. One is we choose sequences that are already unique from the sequence. Second, we computationally filter from based on binding pairs and so on, looking at tendencies. And the third is during the assay, we do a filtering of positive and negative selection. So we select against targets that are similar. So we only retain the ones that are positive.
Okay, thank you for the explanation. And so IDRs, it's a very significant and also very new therapeutic area. So could you share with us on a high level what unique insights that you found throughout your process about the binding behavior of those regions, and how do you want to translate them in therapeutic design?
Sure. I think the lesson learned is, and it's not a surprising lesson, and this time we were more prepared for this, which is it's a challenging problem. It's a very, very challenging problem. It's hard to study IDRs for a reason, but I think we are in the right cusp of time and the right mix of technology to solve this, where synthetic biology has come of age, where you can practically do a lot of these things rapidly, both with next-gen sequencing, DNA synthesis. The read-write has kind of come of age. So if you know how to work with these, very powerful. And then the AI has come of age. Suddenly can reason about sequences and proteins. So I think we're at a cusp of time when this is the start of coming together. So the lesson learned is things are going to be harder than what they appear to be. So plan for the buffer and the time. And there are several aspects that we don't fully understand, right? For example, at the end of the day, the value for the business is how is it creating value? In the drug discovery process, you have to shave some time off or reduce some risk in some way. And that has to have some monetary value. That's how you can claim that you have value of the company. Otherwise, by itself, platforms may not have as much value. And the molecules have to do something. So the molecules that come out are going to be the value generators. So you have to find out, the challenges are find the right disease and area where you have a unique aspect. And there are many there, which is sort of, luckily that's not a problem given this vast space. And then working on aspects that you still don't know. For example, immunogenicity and certain other aspects. So you have to do some expensive assays for those. And then building this whole pipeline where you can sort of screen a very large number of molecules, keep reducing them. And then you have to screen them for properties like solubility, stability, monomeric properties. Produce them in large enough quantities where you can do them in some cell-based assay or sort of send it to some animal-based assay. So we had to build all of this platform literally from scratch. And that itself took time. So you're constantly trying to build resource, raise money to do this, and you're trying to build fundamental aspects as well, and trying to do the science. So it's all happening at the same time, and that's really the major challenge of how do you sort of prioritize rapidly and balance this.
Yeah, so a lot of challenges. And speaking of which, or like maybe not challenges, but let's talk about New Year's resolutions. So the new year started. So what are the resolutions for Aikium for the next year? What are the milestones that you're aiming to achieve given, you said yourself that you always have to have some buffer in time, but maybe something that you want to see happen this year?
Sure. I think we are catching a lot of momentum now. We signed our first pharma partner, and we have a few other partnerships with hospitals. We would like to basically focus on those and maybe close some more partnerships, maybe improve the platform and start sort of cranking. We have all the engine has come together. Now we are sort of cranking it. So increase the design build test cycle pace to rapidly do this, and parallelly also raise the kind of resources needed to fire this at the scale that's needed. Again, having insight of building things in academia and industry, you kind of have a good sense of what it would take to take this all the way through. So you need a certain momentum to build through all of that. So we have a clear sense of what we exactly need to do. What are the milestones we need to hit, the assays that we are working on, and the value inflection points that we are going to reach. So we're planning that we have some buffer. The story is going to be the same for, I guess, any startup, but essentially raise more resource, solve the problems that you said you're going to solve, and continue improving on them, and make sure it's a real problem. So get customers. The reason we are getting partners is not as much even for the dollars that it brings, but this goes to the principle of work with the best people. So only if you work with the best people, Will you understand what is the problem on this ground? Maybe they care about something slightly differently from what you are doing. And we have learned so much from hundreds of conversations that we have had, that we have been constantly evolving. So work with the people where you are going to elevate yourself and solve real problems and demonstrate that those are real problems you're solving. And eventually our goal is, in a few years, we want some of our molecules in the clinic. So from now, that's our goal. We want some molecules in the clinic. How do we get back and sort of hit all of those things?
Yeah, those are great aims. And I hope by the end of the year, you will say that you accomplished all of them or most of them. To wrap things up, because we've been talking a lot about work, about solving great challenges, which is great, but let's focus on the life outside of work. So my final question is, how do you balance the demands of leading a company, solving big problems, spending so much time there with personal fulfillment and with personal life?
Yeah, that's a great question. When you have a startup, a big part of your existence becomes the startup. And it is very important that you have family who support you. So my life is literally family and startup. And I'm fortunate to have a very supportive wife who basically, supports me in doing this. Otherwise, I wouldn't be able to do this. There are parents from both sides that encourage us to do this. It would be seen as a very risky endeavor when you could be getting a normal job, doing things. And I have two kids, 2.5 and 4.5. So it's a very young age. So my biggest fear is missing out on the most important years. But I'm really fortunate because of the support that I get both in family and from my co-founders, that I'm able to balance them. So literally any time is just split between both of them. I organize my life so that my home is relatively close to my startup. So I get all my fulfillment from both of these. So all my hobbies, all my interests are hidden somewhere within there. I love to sketch sometimes, and I sketch ideas. And I get bored, I always doodle. So that's taken care of. I love reading sometimes, and I do read a lot in terms of things that are improving me. There are some great books that come by a recommendation, and I've read that have elevated me as a founder. That I would highly recommend people also, looking at some of these books. Yeah, that's basically it. Just try to. I don't know about balance. I am not a person who understands balance very well. Maybe I understand it a little better now. But take some time for yourself too. Like, I never spent as much time on meditating or doing things. I'm finding that it's very important to have some time to just center yourself every day. It's makes a big difference in the whole day. So those would be my thing. I don't really have something deep and profound to say here. I'm sorry.
No, it was already deep and profound throughout our conversation. So last question. What advice would you give your younger self, maybe 10 years ago, 15 years ago, choose the time?
Great question. I would advise my younger self to doubt myself less, and execute faster. There was an intuition that, hey, I think I can do this. I think I can do this. But there's always this nagging doubt. Really, can you do this? You don't know. You failed your 9th grade. You don't know. Whether you can. You see all the smart people around you who are like so much better and so on. And there's a small doubt that's there. So your pace of execution comes down. My advice to younger self is, be humble, get the advice from people, and just do it. It's okay if it breaks. It's okay if it's failing. But just go do it and doubt yourself less and just build it. It's okay. It's people know as much as they do. And there's no point in doubting yourself. And at the same time, know that you don't know a lot of things, and be very open to advice, and just, and know that you're also not very important in the grand scheme of things. You're not. One guaranteed thing is you have a very limited life. You're born and you're going to die. And of that, a very productive time is going to be very short. And the best way to see this is we all are going to burn ourselves out, but it'll be nice to be an incense stick where in the process of burning, you leave something nice for others to kind of experience through your work or things. Artists leave it in a different way. I would like my life and my work to be doing something positive where I feel like it was worth my life burning out in that sense.
Great insight. So set your ego aside, just go out, build things, solve problems, and create a legacy that goes beyond your own existence and helps people. Thank you very much for the conversation. I really enjoyed it. There were a lot of insights and a lot of lessons for our listeners, and also for everyone who is interested in learning more about how to start a company, the way there. And I think one of the most important, or like at least for me, what I found was one of the most important messages was it's okay to fail. So failure, it's not the end, it's just the start of your journey, and just get up, go on and live your life and go on and solve problems.
Absolutely. Yeah, I think the way I reconcile with that is for one rose, there are 100 thorns. For one shot that comes on TV, or a movie, there are probably 10 times or 100 times more time that's spent. So any good thing that you see, I think there's a feeling that may happen where everybody's doing these cool things, all these things are coming out, but know that there was a very long period that was spent, and the magic happens in the process. So focus on the process, and that naturally failure is part of it. So embracing that keeps me sane and I think that's also what I would tell my younger self. Focus on the process, not just on the results. The process is important.
Very great final words to close up this episode. Thank you very much for joining us.
Thank you so much for this wonderful interview, and it was my pleasure to be part of this. Pleasure and honor. Thank you so much.
Thanks for listening to the Nucleate Podcast. Be sure to subscribe if you'd like to keep up with future episodes. See you next time.